Good morning. My name is Kelly Thomas, and I want to welcome you to our project to provide support for bioinformatics training inside a typical biology curriculum. We're going to do this by sequencing microbes as a way of developing bioinformatics skills while learning some basic principles of microbiology, genetics, ecology, and evolution. The goal of this project is to engage really a large number of undergraduates in this opportunity to have authentic scientific discovery is part of their curriculum and using the tools of modern genetics and bioinformatics. The specific experience that we're talking about providing is allowing students the ability to culture microbes of their choice from the environment of their choice and use the tools of genomics to sequence those genomes, and through that process engage the students not only in their standard microbiology and genetics curriculum, but provide them a substantial introduction to the bioinformatics involved in assembling and annotating genomes. What makes this possible, although these processes have been around for a long time, but what makes them really possible now is this dramatic drop in sequencing costs that have taken place over the last several years and has really transformed many areas of biology. So this is a very commonly used slide from the National Institutes of Health that show the cost of sequencing a megabase of genome over time since 2001, since the first completion of the Human Genome Project. And you can see that there's a period of time around 2007 and really continuing until now where this drop in sequencing costs has been incredible. And the consequence of that is that a class like this, or workshop like this, or an experience like this is really made possible inside of a standard curriculum. So the justification for this particular focus is that when you look across the typical curricula for biology programs at all sorts of different universities and different training environments, the biology core really includes almost always courses in genetics and microbiology, which very often also have laboratories involved with them. And so the bioinformatics for genomics is really a process that uses computer algorithms and that are based on our understanding of core biological processes and core cell biology processes that are part of that curriculum anyway. And so we think this is a really interesting and useful way to both learn basic very important skills in bioinformatics, but at the same time reinforcing the core principles of those disciplines. And so we develop an understanding as we're going through this training in bioinformatics of genome structure and organization, the cell processes of gene expression, such as transcription and translation, and concepts in population genetics and evolution. The obstacles to the development of bioinformatics experiences are really, when it comes down to it, that most faculty in programs like ours and like many across the country, the faculty that are responsible for delivering this information and for engaging the students in these classes are not typically experts in bioinformatics. And the advantage is that for this particular topic is that we typically have lots of expertise at our colleges and universities in the areas of genetics and microbiology. So at least a major part of this experience, we already bring that to our students. The project that we're talking about provides intensive support for the development of core bioinformatics skills. We provide example data sets and tutorials that are similar to real data, and we have a dedicated computer infrastructure that's managed by the New Hampshire IMBRI program. So students and faculty that are at universities that are engaged in this process are not going to be responsible for maintaining their own software and hardware, and will be providing that for you. And there's also online help that is available by appointment to support your courses. Okay, so this curriculum support is being delivered, as you're watching right now, in a very large series of short video presentations, many of which involve hands-on experiences. The curriculum is very flexible. It's meant to be flexible and can be implemented in a whole variety of different contexts, whether it's a long workshop, a section of a course, or a whole semester-long experience. It involves, the first step is the microbiology, so the collect and isolate cultures of bacteria, or phage, if that's your thing of choice, and document and biobanking those samples. The molecular biology part of this can be done by the students in the context of their courses, or it can be done here. We can also provide that as a service, where you need to extract the DNA using standard protocols. And then in response to that, we will sequence the DNA that you provide, and you'll receive a big data file of sequences for each of the students' samples. From there, the students become engaged in understanding and establishing some fundamental skills in bash and command line tools. So these are the basic skills of bioinformatics that are essentially universal for handling any big data projects. And through a series of these tutorials, we will walk the students through the steps of the analysis after a little bit of training, and going through the process of data quality control, genome assembly, genome annotation, and genome comparison as all part of this modular approach. So, I want to recognize the fact that the support for this project comes from the New Hampshire IMBRI program, and for these first several videos, I will be presenting this information to you, and Devin Thomas is providing the editing. In later versions of this, other people will be doing the presentations, and I look forward to working with you.